Hello, everyone. My name is Guangyi Chen, and I am a PhD student in Professor Olga Kalina's lab at Helmholtz Institute for Pharmaceutical Research Saarland in Germany. Today, in this video, I will give a brief overview about how we navigated our way through this year's Canada Challenge. So first, let's start with a basic introduction about why is it important to study antimicrobial resistance, the AMR. In the last few years, the COVID-19 pandemic has had a devastating effect on the lives of millions of people worldwide. But apart from these, there is another invisible pandemic that is spreading silently in the background and affecting many people, and that's the pandemic of AMR. AMR has become an urgent threat to human health worldwide as microbes have developed resistance to even the most advanced drugs. Traditional culture-based antibiotic susceptibility testing, the AST, is still the primary employed method in the clinical labs for assaying AMR in bacteria. Although culture-based methods can accurately detect the AMR, they are resource-intensive in terms of personnel, monetary cost, and time. Further, these culture-based methods are applicable only to a small fraction of bacterial species that are cultivable. For these reasons, the whole genome and metagenome sequencing has become an increasingly prevalent method for AMR characterization. Thus, in this year's Canada Challenge, the goal was to explore the metagenomic surveillance data of about 400 samples provided by the Metasub International Consortium collected across the whole world to trace the AMR patterns. Given the AMR markers in about 150 isolates from the hospital in one of the US cities, the goal was to specifically identify the city of origin of these 150 isolates. Next, we'll look at the pipeline that we used in our study. So the raw reads were first pre-processed with adapter removal to remove remnant adapter sequences and trim low-quality reads. Next, the reads were screened to filter out the contamination from potential host species like humans using Bowtie 2. Filtering out the human sequences is a good practice because all the isolates are handled by humans at some time point, which may introduce some contamination. Finally, the error correction was performed on the non-human reads using base hammer to reduce noise, and these error-corrected reads were then used for further downstream analysis. In the following step, we performed the taxonomic classification directly on the error-corrected non-human reads using Kraken 2. The results are the taxonomic classification report for each sample. We applied the bracket software in the same suit on the report to estimate the relative taxonomic abundance. The results indicate that the three species in the Curie antibiotic-resistant genes, the ARGs, the Enterobacter formicae, Escherichia coli, and Klebsiella pneumoniae, in general, have low relative abundances. Isolates in San Antonio have the highest relative abundance for the Escherichia coli and the Klebsiella pneumoniae. For Enterobacter formicae, New York has the highest relative abundance. After taxonomic profiling, we'll look at the other steps of the pipeline that we use to identify the city of origin for the 150 isolates. First, we did the sample Venn diagram analysis to figure out the city of origin. For this, the error-corrected non-human reads were first used to identify ARGs in the isolates using RGI and Deep ARG. Next, these identified ARGs were pulled together for each of the 6 US cities and then compared with the Curie gene sets using a Venn diagram. The Venn diagram shown here indicates that New York City has the maximum number of ARGs that is in common with the Curie set. So New York City might be the city of origin for the input isolates. However, uneven sampling in this case can be one of the potential issues with this Venn diagram analysis because Minneapolis has only 6 samples, while New York and Denver have 46 samples. To tackle this, in the next step, we did the rarefaction and extrapolation analysis to eliminate the potential uneven sampling effect. It is a data normalization technique that is designed to cope with such unequal sampling effects. By subsampling or upsampling to the same rarefaction or extrapolation depths for all samples. Similar to the Venn diagram analysis, the rarefaction and extrapolation analysis also showed that New York City has the highest number of ARGs that in common with the Curie dataset, and thus the city of origin for the input isolates is New York. Finally, we performed inverse mapping to further confirm our results. For this, the error-correct non-human reads were first assembled using metaspace, and post-assembly, the protein sequences of all the input ARGs were first extracted, and then T plus N was performed to comparison against the assembled context of each sample. After running T plus N, the unique Curie ARGs with at least one significant hit were collected for each city sample, and then the cross-city comparisons was made. The results are shown in this table. From this table, we can observe that over 87% of the input Curie ARGs was found in New York City, while only 74% was found in Denver, which is the next best. This further confirms our claim that the city of origin for the input isolates is New York City. So in conclusion, in this study, we developed the pipeline to explore the metagenomic data and to identify the city of origin for the 150 Curie isolates whose ARG was provided. In the section and rarefaction exploration analysis both showed that the city of origin for the Curie isolates is New York. The inverse mapping further confirms our claim that the city of origin for the Curie isolates is indeed New York. You can scan this QR code for further information. And thank you very much for your attention.